Hello! I am Professor Avery, and today, my little kitties, we are going to learn about the structure of DNA. We all know, or you should know, silly humans, that DNA is what carries all of our hereditary information. Its specific sequence is what makes me a royal feline and you a silly human. We will also talk about RNA briefly, but once you got DNA down, RNA is a piece of cake. Anywho, let us begin by looking at this drawing my mom made of DNA structure.
All right. Now, the first thing you may notice is all these pretty colors. But let's start with these reddish pink and yellow ones. This reddish pink one is called deoxyribose. It is a sugar. It's actually a 5-carbon sugar, 1, 2, 3, 4, 5, known as a pentose. But the name of this specific molecule is deoxyribose, as in deoxyribonucleic acid, or DNA. What connects these deoxyribose sugars are phosphodiester linkages, made up of a phosphate here in the middle, bonded to an oxygen, bonded to a carbon, which is the actual phosphodiester bond. So the backbone of DNA is made up of deoxyribose sugars alternating with phosphodiester linkages. But we want more than just the bones. We want the meat, the good stuff that makes us who we are. So let's look at the nitrogenous bases that actually encode the specific DNA sequence that makes me such a beautiful feline and you such a silly human. When we talk about the specific sequence of DNA, we're talking about the sequence of these bases: adenine, A, thymine, T, guanine, G, and cytosine, C. That's not actually part of the molecule. I just put that there so you humans would understand better. Together, these nitrogenous bases with the deoxyribose sugar and the phosphodiester linkage together make up a nucleotide. So these nitrogenous bases, part of the nucleotides, are split into two categories. We have the purines, adenine and guanine. As you can see, they are 2-ringed molecules in the cooler colors, blue and purple. And you have your pyrimidines, the 1-ringed molecules, cytosine and thymine, in the warmer colors. Now, you may have noticed these little green dotted lines that connect the two molecules together, and these are the hydrogen bonds. The only difference between the G and C bonds is that there are 3 hydrogen bonds, making them a little bit stronger than the connection between A and T, which only have 2 hydrogen bonds. 3 hydrogen bonds, 2 hydrogen bonds. Now, me and my kitty brain are getting a little bored with DNA, so we're going to move over to RNA. Now, one of the things you may notice is that RNA is single-stranded. And there's this new letter, U, for uracil. Uracil, though, is pretty much like thymine, except in that little area. See over here how thymine has an extra methyl group? Uracil does not. Remember what we said before about the ribose in RNA instead of deoxyribose in DNA? So let's look at that again. On this 4th carbon here, we have an OH group, whereas in DNA, we just have a regular hydrogen. So to review, RNA has a ribose with this OH group on the 4th carbon, uracil instead of thymine in DNA, and is single-stranded, whereas DNA is double-stranded. So, to wrap up, we have this double-stranded molecule made of sugars, phosphates, nitrogenous bases. These bases complement each other and make a specific sequence, for example this one, ATG, that codes for who we are. We have RNA, which is very similar, except it is only single-stranded, uses a ribose instead of a deoxyribose, and a uracil in place of thymine. Okay, it is time for me to go now, humans. I am tired, and it is time to nap. Bye-bye.